This is part 2 of the study of eukaryotes. This part 2 will consist of the study of the external structures of eukaryotes, the cytoplasmic membrane, which is the interface between the inner and the outer environment of the cell, and the cytoplasm. Here is a brief overview of part 2. We will look at the external structures of eukaryotes, starting with the appendages or auxiliary organelles, which are the flagella and the cilia. Next, we'll study the capsule and the cell wall, then the cytoplasmic membrane, and finally the cytoplasm. We will mainly focus on structure, composition, and function for each of these items. The flagella is located within the cytoplasmic membrane, and it is an extension of the cytoplasm. The axoneme, shown here as 1 in red, is the inner core cytoskeletal structure. The shaft has tubulin protein. The shaft is shown in a cross section in 5, and the tubulin protein in yellow, and the shape of it is in the form of microtubules. 9 pairs of tubules surround 2 tubules in the center, making a 9+2 structure in the shaft. The basal body, shown as 4 in blue, has 9 triplets of microtubules and none in the center, shown here as 6. And this structure is very similar to that of centrioles. Now, no hook connects the basal body and the shaft, contrary to the prokaryotes, which have a hook in their flagella. In terms of their function, flagella are responsible for locomotion. Shown here are the details of the flagellar structure. An electron micrograph of the cross section of flagella at the levels of the shaft in red and the basal body in blue. At the level of the shaft, there are 9 pairs of microtubules surrounding 2 central microtubules, whereas at the level of the basal body there are 9 triplets of microtubules with none in the middle. Interestingly, the basal body is structurally identical to that of a centriole. Examples of organisms that have flagella are Giardia lamblia, Trichomonas vaginalis, and Trypanosoma cruzi. The cilia are hair-like structures that extend the surface of the cell. They are shorter and more numerous than the flagella. The cilia also has a 9+2 structure of axoneme at the level of the shaft, as shown in this electron micrograph. There are usually hundreds or thousands in number. They beat with rhythm similar to a swimmer doing a butterfly stroke. The cilia aid with clearance of mucus or dirt, and they function by propulsion. Examples of microorganisms with cilia and of cells with cilia are epithelial tissue, which is shown in this top left image, a staining in which you can see clearly the cilia at the top layer of the cell. To the top right is an SEM image of the nasal passage showing cilia. Then a typical microorganism that has cilia is the paramecium. Shown at the bottom left is a light microscopy image, and here is a diagram of the same with details of the internal structures such as the fluid vacuole, the mouth pore, the macronucleus and the micronucleus, contractile vacuole, and the anal pore. The capsule is a sticky structure anchored to the cell membrane via covalent bonds. It has several functions. The main functions are to anchor one cell to another, to strengthen the cell surface, to provide protection against dehydration, and for cell-to-cell recognition. Capsules are absent in eukaryotes that have a cell wall, that is, in plants and fungi. Shown in this electron micrograph is the GLYCOCALYX, that is the capsule of an amoeba. GLYCOCALYX is being shown as GL, the cell membrane as M over here, and then these dark structures are dense arrangement of dense material. So once again shown here is the GLYCOCALYX. Now that we have studied the external structures, let us focus on the cell membrane. The cell wall. The cell wall is composed of various polysaccharides. In plants, the capsule is made up of cellulose. In fungi, it is made up of cellulose, chitin, or glucomannan. In algae, it is made up of cellulose, agar, algin, or silicates. Again, in plants, the cell wall is made up of cellulose. In fungi, of cellulose, chitin, or glucomannan. In algae, cellulose, agar, algin, or silicates. The function of the cell wall is to provide shape and support against osmotic pressure. Shown here is a diagram of the plant cell wall. It shows the cellulose microfibrils in blue, among other structures such as pectin and glycan. Now that we have studied the external structures, we'll focus on the cell membrane. The eukaryotic cell membrane is a fluid mosaic of phospholipids and proteins. It is a bilayer containing steroid lipids, cholesterol in particular, fatty acid chains, and ester linkages. Membrane drafts are a unique feature of eukaryotes, where small distinct regions of lipid and protein molecules that do not mix with other molecules. The membrane drafts serve in cell signaling, protein sorting, and cell movement. The cell membrane has several functions such as passive and active transport, endocytosis, phagocytosis, and exocytosis. Endocytosis involves engulfment of molecules such as proteins. Phagocytosis is the ingestion of cell fragments, microorganisms, or foreign particles, and exocytosis is the process by which the contents of the cell vacuole are released to the exterior. In this diagram of cell membrane, the main characteristics to observe are the phospholipid bilayer, shown here on either side, the presence of glycolipids and glycoproteins, shown here in green and blue, The presence of proteins such as the integral proteins, which span the whole membrane, the peripheral proteins that are on either side of the membrane, as shown here, peripheral, and the protein channels, shown here, that are pore forming proteins that allow ions and molecules to cross in and out by cell diffusion. The eukaryotic cell contains cholesterol, shown here, in the cell membrane. Eukaryotic cell membranes contain fatty acids that are unbranched. When these side chains of fatty acids are added to the glycerol, the organism uses ester linkages in which one oxygen molecule is used to form the link with glycerol, while the others protrude to the sides. And finally, in terms of stoichiometry, it is the D-glycerol that is used in eukaryotic membranes. The cytoplasm consists of the cytosol and the organelles. The cytosol can also be known as intracellular fluid or cytoplasmic matrix. The cytosol is thus the liquid found inside the cells but not inside the organelles. It is also considered to be the complex mixture of substances dissolved in water, including ions, minerals, amino acids, and nucleotides. The cytosol is within the cell membrane, and within the cytosol, cell metabolism takes place. Organelles are internal structures that are either membranous or non-membranous. They include examples such as mitochondria, Golgi, and microfilaments. Many of the organelles carry out metabolic reactions. One important concept to note is that the cytoplasm does not include the cell nucleus. Shown here at the top is a comparison of cytoplasm and cytosol. As you can see from this diagram, the cytoplasm includes everything inside the plasma membrane except the nucleus, whereas the cytosol encompasses all that is inside the plasma membrane except the nucleus and the organelles. At the bottom left, you can see all that a cell consists of a cell membrane, cytoplasm, and nucleus. At the bottom right, you can see that the cytosol is all that is included in the semi-fluid liquid found inside the cell but not inside the nucleus and not inside the organelles. So in summary, we have seen that the flagella and cilia are appendages of the cell that help with locomotion. The flagella are long and few. The cilia are short and many. The capsule has several functions, including avoiding desiccation. It is absent in organisms that have a cell wall. A cell wall is mainly composed of cellulose. It can be found in plants, fungi, and algae only. The cell membrane is a fluid mosaic model. It contains cholesterol and membrane grafts.